Have you ever really thought about what happens after you take a bite of something like a slice of pizza? I mean, the moment you do, this incredible high-speed chemical journey kicks off right inside your body to turn that food into fuel. Okay, so let's dive into this amazing process. We're going to follow a carbohydrate from that pizza crust all the way into your bloodstream.
So that pizza crust, it's absolutely loaded with complex carbohydrates. You can think of them like these massive, tangled necklaces made of tiny sugar beads. They're packed with energy for sure, but they're just way too big for our bodies to use directly. And that structure is actually why complex carbs give you that sustained energy. Your body has to methodically break them down link by link. And that, right there, is the first challenge our digestive system has to solve.
Okay, let's zoom in a little deeper on that crust. We find two main types of these carbohydrate chains. Over on the left, you've got amylose. It's a nice, simple, straight line. Easy enough, right? But on the right, that's its more complicated cousin, amylopectin. See all those branches? That complex, tangled structure is a huge part of what makes a carb complex, and it presents a unique problem that requires a very specialized solution from our body.
So here's the deal. These sugar chains are held together by specific chemical links, and they're called glycosidic bonds. The straight parts of the chain, like in amylose and parts of amylopectin, are linked up by what we call alpha-1,4 bonds. But those tricky branching points you only find in amylopectin, those are held together by alpha-1,6 bonds. To digest these carbs, your body needs a whole set of molecular scissors, we call them enzymes, that are precision engineered to snip these exact links.
All right, our journey begins. The very first place these molecular scissors get to work is, you guessed it, the mouth. The second you start chewing, the chemical breakdown kicks off. Your salivary glands release an enzyme called salivary amylase. This is the first member of the demolition crew. Its specific job? To attack and break those straight-line alpha-1,4 bonds. It gets to work immediately, snipping those long amylose and amylopectin chains into smaller, much more manageable pieces.
But, we don't really chew our food for that long. Salivary amylase only gets a tiny window to work before you swallow, and that food hits the powerful acid in your stomach, which basically shuts it down. So in total, it only accounts for about 10 to 15% of the total carb digestion. It's a clever head start for sure, but our body saves the real firepower for where it's going to be most effective. The main event is still to come.
Okay, so after leaving the stomach, those partially digested carbs make their way into the small intestine. And this is where the real heavy lifting happens, all thanks to a super powerful contribution from the pancreas.
As the food enters the first part of the small intestine, the pancreas just unleashes a flood of its own enzyme, pancreatic amylase. Now, what's really interesting here is that it does the exact same job as the stuff in your saliva, breaking down those alpha-1,4 bonds, but it does it on a massive scale. This is a great example of your body's efficiency. I mean, why waste resources in the mouth when the real work happens later? The pancreas is just the perfect spot for this high-volume assault. So after both of those amylase enzymes are done, we're left with these smaller fragments. You've got maltose, which is 2 sugar units, maltotriose, which is 3, and then these things called alpha-limit dextrins. And this perfectly shows the problem we still have. The amylase enzymes can't break those alpha-1,6 branching bonds, so those are still intact, and these sugars are still too big to be absorbed.
So to tackle these final fragments, our body calls in a team of highly specialized enzymes. They're located right on the wall of the small intestine. This is the finishing crew, the molecular specialists who are going to complete the job.
These specialists are strategically placed on the surface of the microvilli. These are tiny, finger-like folds on the intestinal cells. Now, what's really fascinating here is the design. These folds dramatically increase the surface area of your intestine to roughly the size of a tennis court. It's a brilliant bit of biological engineering that creates this huge workspace to make sure not a single bit of potential energy goes to waste.
And wow, what a team it is. First up, you've got lactase, which breaks down milk sugar. Then there's maltase, handling the maltose we already made. Sucrase is there for any table sugar. See, each one is a specialist. But the real hero of our story, the one we've been waiting for, is isomaltase. It's the only enzyme on this team with the right tool for those tricky alpha-limit dextrins.
Yep, this is its superpower. Isomaltase is the specialist designed specifically to find and break those stubborn alpha-1,6 branching bonds. By dismantling these final branches, it breaks those alpha-limit dextrins down into simple glucose, finally, completing the breakdown of our complex carbohydrates.
Fantastic. So our pizza crust has now been fully converted into single sugar units, glucose, fructose, and galactose.
They're finally small enough to be absorbed. But they still have one last hurdle to cross. They've got to get out of the intestine and into the bloodstream.
And interestingly, they don't all use the same door. Fructose gets a solo pass, entering the intestinal cell pretty easily through a special transporter called GLUT5. But glucose and galactose, they use a buddy system. They actually tag along with sodium to get pulled into the cell through a different transporter, SGLT1. This just shows how the body uses different strategies for efficiency. A simple open door for some and a more energy-intensive buddy system for glucose, the body's main fuel. It's all about smart resource management.
So, while they have different ways of getting into the intestinal cell, all three of these sugars use the exact same exit door to get out and into the bloodstream. This universal doorway is a transporter called GLUT2. It basically shuttles all of our freshly digested fuel out of the cell and onto its final destination.
And with that, the absorption phase is complete. The sugars are now officially in the body. So where do they go to get put to work?
All of these sugars travel through a major blood vessel called the hepatic portal vein, which leads directly to the liver. And the liver, well, it acts like the body's main processing hub, kind of like an air traffic controller. It takes these simple sugars and either sends them out immediately for your cells to use as energy, or it stores them for later. The journey from pizza crust to body fuel is officially complete.
From that first chew to the final exit into the bloodstream, it's just an incredibly efficient and elegant system for creating energy on demand. Every single step is optimized, from maximizing surface area to deploying specialized enzymes exactly where they're needed. But this absolute mastery of turning carbs into fuel leaves us with a pretty fascinating question. What happens when the fuel tank is already full?